I'm Michael Parini. I'm the CEO of Spur Therapeutics. We are a gene therapy company based out of England with presence both in Stevenage, England, as well as in the US in Boston and New York. Really excited about the potential to bring gene therapy beyond rare diseases to more prevalent diseases, but also, really to patients in need with, critical gaps in care today in the rare disease space. And we have a number of clinical stage programs that are designed to hopefully address those gaps. And building on that innovation, we're looking to go after larger patient populations that have unmet need, like Parkinson's disease. So I'm super excited to share a bit about our story. We're very excited about the progress we've made to date in our Gaucher disease program. There's 3 forms of Gaucher disease. We're focused right now on Gaucher type 1, which is the most common form of Gaucher disease. It affects about 18,000 patients in the US, Europe, and Israel. And we've been hard at work developing what we think is a product that can bring enhanced benefits over the treatments that exist today in the form of a one-time treatment. So something that really addresses the unmet need in a way that also frees patients from some of the burdens of care that they experience with their current therapies. We've done a fair amount of preclinical work to get ourselves into human clinical development, which we started not so long ago. We're wrapping up our phase 1/2 clinical trial, where we've had 6 patients participate in the trial. We've completed dosing in that, and the data to date have been incredibly encouraging. For those of you who don't know much about Gaucher, it's a disease that is caused by a mutation in the GBA gene that leads to a deficiency of an enzyme called GCase, which is essential for cellular health. It clears out substrate and byproducts from different cellular activities. And if you don't have this enzyme and you can't break down those substrates, those cells become overburdened and end up either being dysfunctional or dying. And so you end up seeing significant impact from this mutation throughout the body for patients with Gaucher disease. Our approach is to introduce a healthy version of that gene through gene therapy so that patients can make that enzyme for themselves and clear that harmful substrate. And the reason we're so excited about it is we don't just introduce sort of normal gene that produces a normal form of the enzyme, but we introduce a gene that has been invented by our scientists in England, and it produces an enhanced form of the enzyme that is even better at clearing that harmful substrate and hopefully reversing and addressing some of the manifestations of the disease to date. So a very innovative program, and one that the data certainly suggest could be a real game changer for patients with this disease. We certainly put a lot of time and energy into what we call GCase 85, which is the key enzyme in play in Gaucher disease. And early on, we recognized that given the continuous buildup of this harmful substrate in cells, we wanted something that could really address that buildup and do so in a continuous way through gene therapy. And so when we started our Gaucher project, we actually tried to do it with the regular sort of off-the-shelf gene, and we just didn't see a meaningful difference over enzyme replacement therapy, for instance, in the standard of care that exists today. And so when we engineered this new transgene called GCase 85, we were really pleased with what we saw. And certainly the clinical data in Gaucher has played out well, where we see significant clinical improvements in areas that current treatments can't reach. So building on that, and it's well documented in the field that patients who have GBA mutations that may cause Gaucher disease also have a higher risk of Parkinson's disease. And there's been a fair amount of work done to establish the link between GBA mutations in the CNS setting, which cause, again, a deficiency of this GCase enzyme, and the onset of Parkinson's. Frankly, it's earlier in the disease progression. It's a more severe form, and you see patients have a much higher risk of developing Parkinson's if they have this mutation. So with that link established, we felt that we had a real opportunity to change the treatment paradigm for Parkinson's by taking this innovation from Gaucher and carrying it over into that setting. We certainly want to move as quickly as we can because there is a huge unmet need in Parkinson's disease. There certainly are treatments today that are helpful in addressing some of the symptoms, but there are no disease-modifying treatments. And so building on the success in Gaucher and seeing the real difference we've made in patients' lives, we sort of quickly went to work consistent with our strategy of how do we take this innovation from Gaucher and carry it into a different setting. Different organ systems react differently to medicines. We're very sensitive to that. We want to make sure that whatever we introduce into the CNS setting for Parkinson's disease is one, safe, certainly an important topic when you talk about CNS treatments as well as gene therapy, and is most effective in neuronal cells or the cells in the CNS. And so we did a fair amount of work to sort of optimize the product candidate that we wanted to bring forward. And we've completed that work. And now we've selected our lead candidate based on some really exciting preclinical data. And now we're starting to move forward into disease model studies with genetically engineered mice that have Parkinson's disease. And that's a really good way to see if these benefits we're seeing preclinically will translate into improvements in motor and non-motor symptoms in these animals. And so that work is just starting now. We just dosed a number of these mice quite recently, and now we're working quickly and hard to establish a preclinical proof of concept to show it works, and then move it into patients in 2026. Yeah, I think SPR301 is our development candidate for Parkinson's disease. We just recently announced that we're moving it forward in continued development. We're very excited about SPR301 for a number of reasons. First, the preclinical data show us that we have better distribution throughout the CNS. Parkinson's disease affects many of the different cells in the brain, and so getting to all those cells is really important. And we've shown that we could reach all the cells in the brain effectively with a low dose, which is also important in this setting. In addition, we've seen much higher levels of enzyme activity in those cells. So what does that mean? There's more active enzyme to treat the substrate that's accumulating, causing the cell death and Parkinson's disease. So we see really high levels of activity at low doses, which is critical to making sure we can administer treatment that's safe and well tolerated. The last piece that's really exciting for us is we're seeing those reductions in α-synuclein, which is sort of the hallmark of Parkinson's disease, in meaningful ways, well beyond what we've seen others achieve using similar approaches. And that difference is really driven by our unique GCase 85 enzyme. And so it's very exciting for us to see all the things we saw in Gaucher disease and the benefits that GCase 85 confers in that setting translate over into this critical setting where patients really have an unmet need and are all looking forward to having a treatment that actually changes the course of their disease rather than just treating the symptoms.